was it to create amniotic fluid for that womb?
It was challenging. We looked at a lot of research on what constitutes amniotic fluid. We had a lot of dry runs and a lot of hours on the bench trying to produce this. The quantities required for the level of experiments to maintain an animal for 4 weeks requires hundreds of liters of fluid. So even just working out how to make that much fluid in the lab was a challenge. But we have been very gratified that we stuck with it. A lot of earlier research efforts have said that it can't be done. We felt it was really important to achieve fluid incubation in a sterile way. So that is one of the big innovations of this prototype.
You mentioned that inside the lamb would be breathing the fluid. What about nutrition?
So we feed the lambs as well through the circuit. They receive intravenous protein, carbohydrates, and fats, and that's very similar to what the neonates would be given presently in the NICU.
This is the artificial womb facility, a place where humans could be grown entirely from scratch. The devices you see here are called growth pods. Each growth pod is designed to replicate the same conditions that exist inside the mother's uterus. Growth pods are designed to host human fetuses until they are fully developed. These artificial wombs are designed to help premature babies to continue developing after their birth. But emerging scientific research is making it possible to use them to create designer humans entirely from scratch. In the 1990s, researchers at Tokyo University's medical department tested the artificial womb to see if it actually works. They removed a goat fetus from its mother by C-section. Then they placed it in a rubber womb filled with artificial amniotic fluid, and the little guy was delivered 17 days later. In 2002, scientists built mini artificial wombs using cells extracted from the uterus itself. These lab-made wombs allowed embryos to attach themselves to their walls, just like the natural process. Even though the embryos began to grow and develop, they were terminated 5 days later due to ethical concerns. In 2017, scientists from the Children's Hospital of Philadelphia managed to use a primitive design of the artificial womb, where they placed a premature lamb fetus. After keeping the lamb fetus for 4 weeks inside the artificial womb, it started growing a wool coat, gained weight, and even opened its eyes. The researchers went on to test their design of the artificial womb with more lambs, and their experiments were successful.
If you could go back in time and change something about yourself before birth, what would it be? Would you change your eye color, or maybe the color of your hair? Perhaps you'd choose something a little less vain. Maybe you'd boost your resistance to certain illnesses, for example. Well, it's too late for any of us to do this, but for our children, there may be hope. Using gene editing, we may soon be able to design the genes in our babies and select traits we want while discarding those we don't want. The most exciting part? This future may be closer than you think. Until recently, designer babies were a concept of science fiction, something that was only possible in movies. But now, a Chinese scientist claimed that he has created the world's first gene-edited babies. Now that the dawn of designer babies is finally upon us, should we panic or embrace it? The gene editing technique CRISPR-Cas9 has revolutionized humans' ability to edit DNA. But could we take things too far? What if the embryo editing techniques spiral out of control, allowing parents to custom order a designer baby? How would this shape the future of the human race? And at what cost? Only time will tell. Now, it's a bit early to consider ordering from designer baby menu cards to choose your baby's personality and traits in fertility clinics. But the Chinese team used the CRISPR-Cas9 gene editing technique to eliminate a single gene known as the CCR5 in human embryos for the first time. Disabling this gene would make the babies more resistant to HIV, cholera, and smallpox. The CCR5 gene codes for a protein that allows HIV, the AIDS-causing virus, to enter a cell. According to the researchers, embryos were edited for 7 couples affected by HIV, and 1 successful pregnancy led to the birth of twin girls from this treatment. CRISPR-Cas9 gene editing was used to modify the embryos before they were implanted into the respective mother's uterus. Couples could choose whether to use edited or unedited embryos for pregnancy attempts. 11 embryos were used in 6 attempts before the twin pregnancy was achieved. The technique could potentially prevent HIV from being passed on. While the news is causing an outcry in the scientific community, there is no independent confirmation of the claim, and it has not been published in a journal yet. About a year ago, bioethicists did not see the widespread use of CRISPR-Cas9 in the next 2 decades, even for the prevention of genetic diseases, let alone for designer babies. This kind of gene editing is banned in most countries, including the US and the UK, as the technology is still in experimental stages. The DNA changes can pass to future generations, potentially with unforeseen side effects. Genetic modification techniques could lead to the elimination of devastating genetic disorders like Parkinson's disease and Down syndrome before a person is even born, and could help families in which both parents carry genes that increase the risk of a birth defect. The removal of these genes could significantly lower the risk of genetically influenced birth defects, and could eventually lead to the removal of these genes from the entire family tree over time. However, critics warn that interfering with the blueprints of life to prenatally destroy diseases could lead to unintended genetic consequences that are even worse than whatever diseases we're trying to cure. CRISPR-Cas9 has recently been tried in adults as a possible way to cure cancer and treat deadly diseases. But the difference is that these changes are limited to only one person. By prenatally making genetic changes, we take the risk of changing the genetics of a family tree forever. And it's possible that as this technology improves, it will be used for more than just disease prevention. Researchers suggest that even if we focus initially on improving health, it could lead towards non-therapeutic